All right, welcome to this step-by-step explainer. Today, we're diving headfirst into the microscopic world of animal cells and conducting a virtual microscope lab right from wherever you're sitting. So if you've ever found yourself wondering exactly what you're made of on a fundamental level, well, you are definitely in the right place. We're going to break down all this complex biology into some clear, visually guided steps. Let's get into it. To put our topic into perspective right out of the gate, take a look at this number. 100 trillion. That is the estimated number of microscopic cells making up your own body right now. It's an almost unfathomable scale, honestly. But every single one of those tiny, squishy units is working together 24/7 to keep you alive, functioning, and learning. Here's our quick roadmap for today's explainer. We're going to start by meeting your cells, take a peek inside an animal cell, walk through preparing a slide, view those cells up close, compare plant and animal cells side by side, and then wrap the whole thing up with a few review questions.
Okay, section 1. Meet your cells, the absolute building blocks of life.
So, this sounds incredibly simple, right? But it's literally the foundational rule of biology. All animals are made of cells. And because you are an animal, your body is made entirely of these microscopic structures. Now, no one knows the exact, precise count of how many cells are in a human body. It changes constantly anyway. But knowing that you're constructed from roughly 100 trillion of these tiny biological blocks makes understanding them pretty crucial to understanding your own existence. I mean, they are the baseline of literally everything you do. Moving right along to section 2, inside the animal cell, exploring those organelles. Let's map out the 4 shared components you're always going to find inside an animal cell. If you picture the cell's anatomy, you've got that outer boundary, that's the cell membrane. Inside that boundary, there's the cytoplasm, basically filling up all the empty space. Then you'll notice the mitochondria. They usually look like little oval shapes. And finally, the large, very distinct nucleus. These are the fundamental working parts that just keep the whole operation running smoothly. Now, to make this stick, let's use some simple analogies. It really helps. You can think of the nucleus as the cell's headquarters, basically calling the shots and directing operations. The cell membrane? That acts like a security gate, constantly checking and controlling what gets to go in and out of the cell. The mitochondrion is essentially the power plant. It provides the energy the cell needs to actually do its job. And finally, the cytoplasm is kind of like this jelly floor that fills the space where everything inside the cell takes place. Keep these comparisons in the back of your mind, and visualizing the cell's internal machinery becomes a total breeze. All right, section 3, preparing your microscope slide, a step-by-step guide. Before we actually start swabbing for a sample, we've got to talk about our tools, specifically something called a stain. A stain, like methylene blue, is a dye we use for a very practical reason. Animal cells are incredibly small and naturally transparent. If you just slapped a plain cheek cell under a microscope, you wouldn't see much of anything. So the methylene blue acts as a dye. It turns those transparent parts a dark blue color, which makes them pop and become easily visible to the human eye under a lens. Let's walk through the actual lab procedure here. Step 1, safety always comes first, so throw on your safety glasses. Step 2, very gently rub a cotton bud along the inside of your cheek. This simple action collects some loose cheek cells. Next, for step 3, you just rub that cotton bud right onto the surface of a clean microscope slide. Remember, you can't see them yet, they're tiny and clear. Step 4 is where the magic happens. Use a dropper to add a single drop of that methylene blue stain right onto the cells. And finally, step 5, carefully lower a small glass cover slip over the drop of blue stain. Bam! Your slide is completely prepped and ready for the microscope. Which brings us to section 4, viewing cells up close, focusing the microscope. Operating a microscope is all about a careful, really systematic approach. First off, you always want to start by clicking the smallest objective lens into position directly over the stage. Second, place your freshly prepared slide onto the stage and make sure the sample is right over the hole where the light shines through. Third, and this is key, look from the side, not through the eyepiece yet, and turn the focusing knob until the lens gets super close to the slide. Finally, step 4, now you look down the eyepiece. Slowly turn the focusing knob to move the lens upwards until those stained cells pop into a crisp, sharp focus. Once you've mastered finding it with a small lens, then you can switch to a larger one for a more magnified view. And boom, this is the payoff. This brilliantly illustrates the beautiful, complex structures you'll discover once you get your microscope perfectly focused. Thanks to that methylene blue dye we added, the normally invisible outer borders and those dark nuclei right at the center of each cell just stand out spectacularly against the background. It truly is like a whole hidden universe, sitting right inside your mouth. Next up, Section 5, Plant versus Animal Cells. Let's spot the difference. Check out this visual comparison. On the left side, we've got a magnified view of plant cells. Notice how they look almost like a brick wall? They are super tightly packed, very geometric, and incredibly rigid. Now look over at the cheek cells on the right, the animal cells we just learned how to stain blue. Totally different vibe, right? They're scattered, kind of rounded, and have much softer, irregular edges. By analyzing these side by side, we can instantly spot some pretty stark visual differences in their overall shape and structure.
So, why the huge visual difference? Well, animal cells have those rounded, irregular shapes, and they rely on the four key parts we talked about earlier: the membranes, cytoplasm, nucleus, and mitochondria. Plant cells, on the other hand, form those rigid, block-like structures. Now, they actually do contain those exact same four base organelles as animal cells, but they have specific extra pieces, like a really tough outer cell wall. Those extra pieces are absolutely necessary for plant life, and they're exactly what gives plant cells that stiff, brick-like appearance compared to the much softer, squishier animal cells. All right, final stretch. Section 6, student review questions. Time to test your knowledge.
Okay, pop quiz time. Think back to the tutorial steps we just covered. Do you feel totally confident knowing how to prepare a slide using a cheek swab and some methylene blue? And do you remember the safe way to place that slide on the stage and focus the smallest objective lens upwards? If you can clearly visualize those steps in your head, you are well on your way to mastering basic microscopy. Moving to question 2. Take a quick second to recall those simple analogies we used. Remember, both plant and animal cells share the headquarters nucleus, the security gate membrane, the power plant mitochondria, and the jelly floor cytoplasm. But don't forget the distinct visual differences we spotted under the lens, the kind of scattered, irregular shapes of the animal cells versus those highly structured, rigid blocks of the plant cells. And finally, question 3. Let's say you are handed a mystery photograph right now of some cells viewed under a microscope. What specific structural features would you look for to solve the case? Well, you'd immediately check out the borders, right? If they look like a soft, scattered mosaic, you're almost certainly looking at an animal cell. But if they look like a sturdy, highly organized brick wall, yep, you've got a plant cell on your hands. I'm going to leave you with this final provocative thought to chew on for your future studies. If both we and the plants outside our windows are built from fundamentally similar microscopic building blocks, we both share a nucleus, a membrane, a cytoplasm, all of that, then what are the hidden, highly specialized instructions inside those cells that make us so wildly, fundamentally different? Keep asking those questions, keep looking closely at the world around you, and we'll catch you in the next explainer.